Welcome back everyone. So in today's lecture we are going to learn about Epstein-Barr virus, which is one of the members of herpes viridae family, a group of viruses which are having double-stranded DNA. And this Epstein-Barr virus causes an infection called as infectious mononucleosis, and it is also associated with the several human tumors like nasopharyngeal carcinoma, Burkitt's lymphoma, Hodgkin's disease, B-cell lymphoma, etc. So this Epstein-Barr virus is the first known virus which is associated with human carcinomas, and it is also coming under oncogenic viruses. So coming to morphology of Epstein-Barr virus. So this Epstein-Barr virus belongs to gamma subfamily of herpes viridae. So there is a family, herpes viridae family. Under that there are three subfamilies. One is alpha, another one is beta, and one more is gamma. So this Epstein-Barr virus belongs to gamma subfamily of herpes viridae. And all these herpes viruses which belongs to alpha, beta, gamma, all these are having double-stranded DNA. They are enveloped and the nucleocapsid is having icosahedral symmetry. This is the structure is same for all the herpes viruses. And one more point about Epstein-Barr virus is that it expresses three classes of antigens. So one is latent phase antigen, another one is early antigens, and one more is late antigens. So all these antigens serves different functions. So these late antigens, they are the structural proteins. They form the structure of virus. They form the viral capsid and envelope. And early antigen, this is a non-structural protein which mainly helps in the pathogenesis of the virus. This help in the viral replication inside the host cell. And third one is latent phase antigen. They are mainly synthesized during period of latency. So all these herpes viruses have a special characteristic feature of undergoing latency. So initially they will cause primary infection in the host, then they will undergo latency for several period of time, maybe up to months to years, and again during certain stimuli they will reactivate, causing latent infection. So in Epstein-Barr virus, these latent phase antigens are synthesized in the B lymphocytes after primary infection during the period of latency, and it results in further complication. So coming to pathogenesis of Epstein-Barr virus. So this mode of transmission of Epstein-Barr virus is oropharyngeal contact through the salivary secretions. Hence the infectious mononucleosis. Which is an infection caused by Epstein-Barr virus, is also called as kissing disease or glandular disease. Since the mode of transmission is oropharyngeal contact through salivary secretions, this infection is mainly seen in younger adults. Maybe during kissing, this virus will spread from one person to another person. Hence it is called as kissing disease. And it is also called as glandular disease because this virus enters through the mouth, right? So it multiplies in the oropharynx, in the salivary glands. Hence it is also called as glandular disease. So let's move on to the pathogenesis of this virus. How does this virus causes infection in the host? So since the mode of transmission is through oropharyngeal contact, the virus enters into the mouth. Initially virus enter into the mouth and binds to the specific receptors present on the B cells and pharyngeal epithelial cells, that is CD21. So CD21 receptor of B cell and pharyngeal epithelial cell. So before invading into the host cell, the virus has to attach to the receptor. So this virus attaches to the CD21 receptor of B cell and pharyngeal epithelial cells. After attachment, it replicates inside the epithelial cells or surface of the B lymphocytes of the pharynx and salivary glands. So continuous multiplication of the virus is going to take place. After that, these infected B cell, the B cells become infected. Instead of destruction of B cell, this virus makes these B cells immortalized, which means B cell continuously, this infected B cell continuously going to divide. As a result, it will synthesize large number of variety of antibodies or immunoglobulins. So large number of polyclonal immunoglobulins will be synthesized. So many of those will be autoantibodies, which destroys the self antigens. As a result of this synthesis of polyclonal antibodies by the B cell, there will be stimulation of CD8 T lymphocytes. To clear that infected B cell, the CD8 T lymphocytes will be stimulated in large number. As a result, it will appear atypical. So, atypical lymphocytosis. This is one of the very important observation or the finding in Epstein-Barr virus infection. Atypical lymphocytes will be prominently seen. And next, after causing primary infection, this virus as I told, this virus undergoes latency in infected B cells, right? During this period, these latent phase antigens will be synthesized. These latent, like LMP and EBNA. So, late membrane protein and Epstein-Barr nuclear antigen, these will be synthesized, which result in malignant transformation of the infected B cell. As a result, there will be carcinogenic property in this virus. So there will be continuous multiplication as well as malignant transformation of the infected B cells, which turns into tumors. Then coming to clinical manifestations, as I have already told, this Epstein-Barr virus causes infectious mononucleosis, which is also called as Kissing's disease or glandular disease. So the prominent symptoms in this infection will be headache, fever, malaise, pharyngitis, cervical lymphadenopathy, hepatosplenomegaly, rashes following ampicillin therapy, and atypical lymphocytosis, which is one of the important finding, and autoantibodies, which are reactive to sheep RBCs. Then about this we have already discussed. There are several carcinomas which are associated with Epstein-Barr virus infection. Then other conditions. Sometimes there will be lymphoproliferative disorder in the person as a result of continuous multiplication of the infected B cell and malignant transformation of infected B cell. There will be high number of lymphocytes, that is B lymphocytes in the blood. And oral hairy leukopenia, you can see in this picture, there will be white hair-like patches on the tongue, which are non-removable, patchy white hair-like things will be there on the tongue. And chronic fatigue syndrome. So in this patients, there will be weakness or fatigue which is for long period of time, maybe more than 6 months. Then lab diagnosis, specimen that can be collected will be serum sample, antibody detection. There is one important heterophile agglutination test specifically for Epstein-Barr virus infection antibody test, that is Paul Bonnell test, which is a heterophile agglutination test. Then moving to confirmatory test. Result of Paul Bunnell test can be mainly confirmed by monospot test or differential absorption test. Then other methods includes ELISA and indirect immunofluorescence. Molecular method to detect DNA will be PCR and real-time PCR. Then virus isolation can also be performed, but in the routine diagnosis this is not performed since it is a prolonged procedure and it is a time consuming one and requires experienced personnel. This is not preferred. Thank you.